In our previous presentation, we looked at the four kingdoms found in eukaryotic domain. Now we will look at the two prokaryotic domains. The two prokaryotic domains are the domain archaea and the domain bacteria. These two domains have some similarities in that organisms found in both do not have a nucleus to contain their genome. Genome is just all of the chromosomes that code for the genetic material of a particular organism. These two domains also, their cells do not contain membrane-bound organelles. If you think of the cell as a factory, an organelle is a little machine that has a specific function. The first living organisms found on the planet Earth were prokaryotes. There's evidence of their existence found in rocks dated to be about 3.5 billion years old. The conditions on the Earth at this time were very different than they are today. The temperatures were very high, and there was very little free oxygen. Organisms in the domain bacteria are the most diverse and prevalent organisms on our planet. There are tens of thousands of different types of bacteria being identified, and there's probably many more that exist. In identifying a bacteria, one of the things that's commonly looked at is the structure or the shape. Bacteria exist in three common shapes. They can be long, thin rods or bacilli. Circular bacteria are characterized as being spheres or cocci, and they can kind of be corkscrew shaped, spiralia or spirochetes. Sometimes these organisms have a particular arrangement. For instance, there are cocci that when they replicate, they kind of remain attached to each other, and they form cocci. They form chains of cocci or clusters. If we look at the domain archaea, the structure of the plasma membranes, this is the outside boundary of the cell, contains very unusual fats or lipids. And this allows these organisms to function at high temperatures, temperatures that would kill other organisms, including the members of the domain bacteria. What type of archaea are the methanogens? And these are organisms that produce methane gas, and they couple this with the production of ATP. ATP is the currency of energy that is used by all living cells. Methanogens can be found in swamps as well as the intestines of animals. Halophiles are organisms that require very high salt concentrations to grow. So what was once known as the Dead Sea, dead because it's devoid of any living plants or marine life, actually does contain some bacteria. There are some members of the domain archaea that are categorized as being thermoacidophiles. That means they can grow at very high acidic conditions. The suffix philes or philic at the end of the term means loving. So if you look at the word thermoacidophiles, it means organisms that love very hot acidic temperatures. They grow best at temperatures above 80 degrees C. Keep in mind that water boils at 100 degrees C, so this is very hot indeed, and at a pH about 1 or 2. One member of this group is Thermus aquaticus, and this was first discovered in the Yellowstone National Park hot springs in the 1960s. These organisms, again, can grow at very high temperatures, and there's enzymes that are necessary for the replication of DNA before an organism splits into two progeny. And so what this enzyme can be used for is in order to make copies of human DNA molecules. Okay, so at a crime scene, a very small quantity of DNA may be isolated. And in order to do DNA fingerprinting, this DNA needs to be replicated many times. The enzymes inside our body will not function at a high temperature, though a high temperature is necessary to have the double-stranded DNA molecule separate. And so DNA strands can be heated to induce separation, and then the enzymes from this bacteria added to make copies of these molecules. This process was finalized in the late 1980s, and so in 1989, the Science Molecule of the Year was awarded to the enzymes involved in the PCR reaction as isolated from the Thermus aquaticus bacteria. So we mentioned when we were talking about the plant kingdom, the drug Paxil, which is isolated from the bark of Pacific yew trees. And this drug, though synthetically manufactured now, can be used to treat ovarian and breast cancer. Biodiversity describes the variety of life on Earth, in regards to the number of different species. On our planet, there's about 15 million species, and less than 2 million of these have been classified. If a species go extinct, that means the entire species or taxonomical group dies. It's estimated that about 400 species go extinct every day due to human activity. If we continue to allow these species to become extinct, we may be losing our next vital drugs that could be used to treat other cancers or other types of diseases found in humans. If you looked at a single teaspoon of soil, it might contain up to 1 billion microorganisms, each of these microorganisms possessing the characteristics of life. The number of bacteria on your skin is roughly equal to the number of humans on the planet Earth.